Hello, dear scientist-to-be. Welcome back to the Geneticist. In our last episode of How to Biotech Laboratory, we met the Blotting family. Now, as promised, we're doing a deep dive into the most famous and widely used member, the Western blot. Today, we're going on the bench for a comprehensive step-by-step guide, covering everything from core protocols to advanced optimizations and troubleshooting. And we're back. The Western blot is a core technique in molecular biology and biomedical research used to detect, identify, and quantify specific proteins within complex mixtures. It's valued for its specificity, sensitivity, and ability to provide information about a protein's size and abundance. So why do we use it? 1. Protein identification. It allows researchers to confirm the presence of a specific protein in a sample, even among thousands of others. 2. Quantification. We can estimate the relative amount of a protein to compare between different samples, like healthy versus diseased tissue. 3. Protein characterization. It provides details about a protein's molecular weight and post-translational modifications, such as glycosylation. 4. Clinical and research applications. Western blotting is used in diagnostics, like HIV testing, and in a wide range of basic science to study protein function and expression. The entire workflow typically starts with protein extraction and separation using SDS page, then the blotting itself, and finally, data analysis. Okay, let's get our lab coats on and walk through the core protocol from start to finish. Step 1. Sample preparation and gel electrophoresis. First, we prepare our samples by extracting proteins from cells or tissues. Then we quantify them to ensure we load an equal amount of protein into each well. Next, we load these samples onto an SDS page gel to separate the proteins by their molecular weight. We also use appropriate running and sample buffers for optimal separation. Step 2. Protein transfer and membrane selection. Once the proteins are separated, we transfer them from the gel to a solid membrane using electrophoretic transfer. The membrane is crucial. You have two main choices, PVDF or nitrocellulose. PVDF membranes are more suitable for multiple rounds of antibody probing, reprobing, while nitrocellulose membranes may offer higher sensitivity, especially for low-abundance proteins, and are often easier to handle. Step 3. Blocking, antibody incubation, and detection. This is the heart of the Western blot. Blocking. We first incubate our membrane with a blocking buffer to prevent non-specific binding of the antibodies. Primary antibody. We then incubate with a specific primary antibody that is designed to bind only to our target protein. Secondary antibody. After washing away any unbound primary antibody, we add a labeled secondary antibody. This one binds to the primary antibody and carries a label that lets us see our protein. Detection. Finally, we visualize the bands using methods like chemiluminescence, colorimetric, or fluorescent detection. This process provides our final visual result. Even in the best labs, things don't always go perfectly. Knowing how to troubleshoot common issues is a key lab skill. 1. Weak or no signal. This could be due to low protein or antibody concentration or transfer issues. To fix it, you can increase your protein or antibody concentration and optimize your transfer. 2. High background. This often means inadequate blocking or too much antibody was used. To solve this, improve your blocking step or dilute your antibodies. 3. Non-specific bands. If you see extra bands, your antibody might not be specific enough. The solution is to use validated antibodies and optimize your washing steps to get rid of the junk. 4. Uneven bands. This can come from gel or transfer issues, so make sure your gel is polymerized evenly and the transfer is consistent. Troubleshooting requires careful attention to each step, from sample preparation to detection, and is key to obtaining clear, reproducible results. The Western blot is a classic, but scientists are always making it smarter. Optimizing the protocol is crucial for improving its sensitivity and reproducibility. Key protocol optimizations. 1. Streamlining the process. We can now make gels in just 10 minutes and run electrophoresis in as little as 35 minutes. Using ethanol instead of methanol and certain blocking agents can cut down the total time to about 80 minutes. And performing washes at 4 degrees Celsius can actually maximize the signal-to-noise ratio. 2. Improving sensitivity. You can fix proteins to the membrane using a combination of organic solvents and heat to prevent protein loss, which can increase the signal up to 16-fold. And for some proteins, denaturing them on the membrane after transfer can dramatically improve antibody accessibility. 3. Membrane choice. The choice of membrane really matters. Nitrocellulose membranes can provide higher sensitivity for some proteins, especially when combined with a denaturation step, which can increase detection sensitivity by over 100-fold for certain targets. The future of Western blotting is even more exciting, with new technologies pushing the boundaries of what's possible. Advanced methods. Innovations like single-cell Western blotting, microfluidic Western blots, and automated systems increase sensitivity, reduce errors, and allow for high-throughput analysis. Microfluidic Western blotting, for example, can give you rapid, highly sensitive, and multiplexed protein detection in just 10 to 60 minutes. Next-generation detection. We're no longer just using chemiluminescence. New methods include SIRS-based Western blot. This uses nanoparticles to achieve detection limits as low as 0.15 picograms. In-gel fluorescence. This antibody-free method offers higher sensitivity and better signal-to-noise ratios than traditional Westerns. Proximity ligation. This technique uses dual antibodies and amplification to increase sensitivity by at least 16-fold and improve specificity. These key improvements, from faster protocols to new detection methods, are making Western blotting more reproducible and quantitative than ever before. Wow! Western blotting is a core technique that continues to evolve, from its foundational principles to new, advanced methods that are pushing the boundaries of sensitivity and specificity. In 2025, it remains the most widely used blotting technique in research and clinical labs due to its versatility and ongoing technological improvements. So, if you're as fascinated by the amazing world of biotech as we are, hit that subscribe button right now. And don't forget to ring the notification bell so you never miss a single revolutionary discovery from the Geneticist. This episode is just one part of our exciting How to Biotech Lab playlist, so dive deeper there for more incredible insights. And for all the detailed research and citations we mentioned today, you'll find them conveniently in our Telegram channel. All the links you need are in the description below. See you in the next episode inside the amazing world of the cell.